न्यूक्लियस स्टार्ट करथिला तो न्यूक्लियस रो किछटा पार्ट हमें डिस्कस करथिला। तो न्यूक्लियस अंडर रे हमें डिस्कस करथिला डिफरेंट कंपोनेंट्स एंड लास्ट क्लास रे डिफरेंट कंपोनेंट्स रे जेंटा के हमर कंप्लीट हेतिला न्यूक्लियस अंडर रे हमें पढ़िथिला न्यूक्लियर मेम्ब्रेन, देन न्यूक्लियर मेम्ब्रेन पर हमें देखिथिला तार भितरे न्यूक्लियस रे भितरे रहउछि तार न्यूक्लियोप्लाज्म एंड न्यूक्लियोप्लाज्म जेंटा कि तार सेमी लिक्विड पार्ट टा कहउछु न्यूक्लियस रे जेंटा सेमी लिक्विड पार्ट जेंटा कि कहउछु न्यूक्लियोप्लाज्म बोली करी एंड न्यूक्लियोप्लाज्म पर हमर रहउछि नेक्स्ट पार्ट जेंटा कि कहउछु हम क्रोमाटिन मटेरियल बोली करी कहउछु जेंटा कि आज डिस्कस करमा जेंटा कि कहउछु क्रोमाटिन मटेरियल एंड क्रोमाटिन मटेरियल कहा के नेगिर बने छे दुईटा जिनिस के नेगिर बने छे। दिस इज मेड अप ऑफ टू थिंग्स डीएनए एंड हिस्टोन प्रोटीन एंड क्रोमाटिन मटेरियल टा परे दिस इज कन्वर्टेड इनटू क्रोमोसोम। क्रोमाटिन टा कन्वर्ट हो जाम क्रोमोसोम लिके देन अनदर पार्ट जेंडा डिस्कस करमा दैट इज योर न्यूक्लियोलस पार्ट। तो एतकी पार्ट डिस्कस करबार अच्छे तो एतकी भीतर हम ऑलरेडी डिस्कस करी सारी जो न्यूक्लियर मेम्ब्रेन एंड न्यूक्लियोप्लाज्म। देन लेट्स कम्स टू द क्रोमाटिन मटेरियल पार्ट। तो क्रोमाटिन मटेरियल कहा के कहूँ इफ यू सी chromatin material chromatin material काना कहूँ दिस इज ए थ्रेड like elongated structure. This is a thread like elongated structure and I am last class पढ़िथिला जे chromatin material टा this is found in the interphase. This is found in interphase. Last class रे online class रे discuss करथिला interphase रे this is seen. Interphase रे काना होचे chromatin material टा देखबार के मिलु छे जेनथिरे कि हमर काना रहउचे जेनथिरे कि क्रोमाटिन मटेरियल रा हमर डीएनए टा कॉइल्ड होउचे कहार अराउंड रे हिस्टोन प्रोटीन अराउंड रे कॉइल होउचे। ओके सो इंटरफेस टा कहा के कहउछु इंटरफेस होउचे हमर जते बेले सेल टा डिवाइड नेवर व्हेन द सेल इज नॉट इन डिवाइडिंग कंडीशन तो एटा के कहउछु हम रेस्टिंग फेज बोली करी कहउछु। दिस इज द रेस्टिंग फेज वेयर द सेल विल नॉट डिवाइड हियर द सेल विल नॉट डिवाइड जेन फेज रे सेल रो डिवीजन न हेबार से फेज के कहउछु इंटरफेज। तो इंटरफेज कंडीशन रे हाउ दिस डीएनए विल लुक सपोज दिस रेड पार्ट दैट इज द डीएनए सपोज इटा हला हमर डीएनए काना करबा इट विल कॉइल अराउंड द न्यूक्लियोसोम और दैट विल कॉइल अराउंड योर हिस्टोन प्रोटीन। सपोज दिस इज हिस्टोन प्रोटीन दिस इज अनदर हिस्टोन प्रोटीन दिस इज अनदर हिस्टोन प्रोटीन दिस इज अनदर हिस्टोन प्रोटीन सपोज। तो हिस्टोन प्रोटीन रो अराउंड रे डीएनए विल कॉइल इधर अराउंड रे डीएनए रो काना हवा कॉइलिंग हवा अगेन इट विल कम्स टू कॉइल विथ द हिस्टोन प्रोटीन अगेन इट विल कॉइल एंड अल्टीमेटली देयर विल बी फॉर्मेशन ऑफ चेन ऑफ हिस्टोन प्रोटीन एंड डीएनए। दिस इज द हिस्टोन प्रोटीन दिस ब्लू कलर पार्ट that is the histone protein and this red part that is the DNA. So DNA when coil around the histone protein it will make what? It will make the when the DNA this is the DNA coil around the histone protein and it will make the chromatin material. This will make the chromatin material. Okay, what is chromatin material? Chromatin material is the chain of DNA with the histone protein. When the number of histone protein they will make, we'll discuss that how this chromatin material will form. Okay, so before that, just we have to know what is that chromatin material. Chromatin material is nothing but that is having the DNA and the histone protein. And when this coiling and recoiling will occur after several coiling and recoiling in this part, that will make the compact structure. We call this structure as the chromosome. Chromosome kaha ke kohuchu? Chromosome is the condensed form, or when this chromatin material undergoes repeated coiling and recoiling. When the chromatin material undergoes repeated coiling and recoiling, it will make the chromosome. This chromosome will form when this chromatin material will undergo the process of repeated coiling and recoiling. This will undergoes repeated coiling and recoiling. Okay, so when this chromatin material undergoes coiling and recoiling, that will make the compact structure, and that compact structure we call this structure as the chromosome. Okay, so let's see how this condensation process or how this coiling and recoiling will occur, and that ultimately results in the formation of our chromatin material. And that chromatin material, when undergoes repeated coiling and recoiling, that will make the chromosome. So that process is known as your DNA packaging process. Okay, so in a normal cell, suppose this is one cell. In a normal cell, suppose this is one cell. In a normal cell, there is presence of a nucleus. Here suppose this is the nucleus. So inside the nucleus, the chromatin material is present. Suppose this is the DNA. This is one DNA. This is another DNA. So here the DNA that exists in the form of thread. So this material, that is called as chromatin material. This is called as chromatin material. So chromatin material will found this chromatin material in which phase? That will found in the interphase, when the cell is not dividing. Okay, but when the cell will undergoes division, at that situation जेतेबेले cell रो division हवा, division हला time रे जदि हमें chromatin material के देखमा, then the chromatin material or that DNA undergoes several coiling and recoiling, and that will make the chromosome that will exist in this form. In the cell divide, जेतेबेले cell रो division होचे, during the time of cell division, that will seen in this stage. That means this is the compact form of the DNA. Here this structure that is made up of what? This structure that is made up of nothing but your this part, all these two part, that is your chromosome. But these chromosome are made up from what? That is made up from your this chromatin material. When this chromatin material undergoes several coiling and recoiling, they make the structure that is called as your chromosome. So this is the chromosome. This part, this is the chromosome. So chromosome ken time re dekhma? The chromosome that will found in the mitotic phase. In the mitotic phase. What is mitotic phase? This is the cell dividing phase. जेतेबेले cell रो division होचे से phase के कहूँ हमें mitotic phase बोली करी. And cell जेतेबेले divide नहीं हबार से phase के कहूँ हमें काम बोली करी interphase बोली करी कहूँ. तो interphase रे the chromatin material will be found, but in the mitotic phase the chromosome will found. Okay, that means this chromatin ultimately that results in the formation of your chromosome. Okay. So let's see how this process of condensation occur, or how this process of coiling and recoiling occur, and that make the chromosome. Okay, write this. Okay, I will share this. Let's see. DNA packaging. What is DNA packaging? DNA packaging means in a cell. Why that DNA packaging would require? If you see one cell have the DNA, one cell have the DNA, but the length of this DNA, is much greater. This is much greater. Okay, the length of the DNA that is much greater as compared to the cell. Good cell ro size jete bole dekhu cell ro size ru DNA ro length jodi dekhma, that is बहुत high, that is much greater than the size of the cell. So this is, and in that cell there is presence of nucleus. In that cell there is presence of nucleus, and inside that nucleus, inside the nucleus there is presence of our DNA. Okay, so as the DNA is very long, that DNA require what? That DNA require packaging. Okay, that DNA undergoes coiling, and that coiling will make this DNA to fit inside the nucleus. गोटे न्यूक्लियस भितर फिट होकर रहबार लगी डीएनए के काना दरकार होचे? डीएनए रिक्वायर कॉइलिंग एंड रिकॉइलिंग, एंड दैट प्रोसेस ऑफ कॉइलिंग एंड रिकॉइलिंग of DNA, that process is known as what? DNA packaging. So what is DNA packaging? The process of, this is the process of coiling and recoiling of DNA, coiling and recoiling of DNA to fit inside the nucleus of the cell. That is called as what? It's called as DNA packaging. Okay, so when the DNA will undergoes packaging, first of all, what will happen? DNA, from the DNA there will be formation of chromatin, and that chromatin will make, ultimately when that undergoes after several coiling and recoiling, that will make the chromosome. And this whole process that is known as what? That is we called the DNA packaging. This is called as DNA packaging. Okay, so let's see the mechanism of this DNA packaging. Okay, okay, let's see. Mechanism. Mechanism of DNA packaging. So for the DNA packaging, what is required? What is the requirements of this DNA packaging? So when we see for the DNA packaging, there will be requirement of a protein, and that protein, that is called as histone protein. That protein is the histone protein. Okay, so when we see this histone protein, the histone protein are of 5 types. These are of 5 types. One is the H1 histone protein. Okay, so one is the H1 histone protein, then another one H2A, H2B, H3 and H4. These are the 5 types of histone protein that required for the DNA packaging. So these are the 5 types of histone protein. So out of this histone protein, let's draw this in a graphical manner. All this protein, let's see all this protein in a graphical manner. Suppose this is the H1 histone protein. Then there will be your H2A, then H2B, H3, and the last one, that is the H4. Okay. So out of this H1, H2A, H2B, H3 and H4 type of histone protein, from the H2A, H2B, H3 and H4, from this each molecule, out of this four molecules, 2 molecules of H2A, H2B, H3 and H4. जेतेबेले two molecules of H2A, H2B, H3 and H4, they will fuse, they will make a 8 molecular structure. They will make a 8 molecular structure. Okay, जेतेबेले दुईटा molecules of H2A रो दुईटा, H2B रो दुईटा, H3 रो दुईटा and H4 रो दुईटा molecule जेतेबेले they will fuse, they will make a 8 molecular structure, and this 8 molecular structure is called as octamer. Octa means 8, that's why that is called as, that 8 molecular structure is called as octamer. So in this octamer there will be presence of, in this octamer there will be presence of our all these histone protein H2A. Suppose this is H2A, one H2A is there, this is another H2A, then 2 molecule of H2B, this is another H2B, then 2 molecules of H3, and 2 molecules of H4. Okay, so this structure which is formed here having 2 molecule of H2A, 2 molecule of H2B, 2 molecule of H3 and 2 molecule of H4. And this structure that is known as what? This is called as octamer. Okay. And around this octamer what will happen? Around this octamer the DNA will comes to bind and it will coil around the octamer. Octamer around रे काना हवा हमर DNA आसिकरी काना करबा DNA will comes to coiled around the octamer. Suppose this is the DNA. The DNA will come. Suppose this is the DNA. The DNA will comes to coiled around the octamer and it will again release. DNA is very long. Okay, but here the DNA only coiled, it will only coil about 1.75 ton. DNA काना करबा 1.75 ton. 1.75 ton काहे के कहूछु देखा DNA गोटे full ton, DNA गोटे full complete coiling कला. This is one complete coiling. Then second coiling जते बेला होचे, second coiling that is not fully complete. Second coiling टे कना हवा गोटे full coiling हवा and second coiling केते हवा 3/4 हवा, 4 भाग रो 3 भाग. Okay, so this is called as 1.75 ton. DNA will coiled दुईटा पूरा full coiling ना हे करे गोटे coiling टा full हवा and आराटा 3/4 coiling हवा तो सता के कहूछु 1.75 ton not 1.75 coiling. So the DNA will coil around this octamer and ultimately after 1.75 coiling it will again remove, and if there is presence of another octamer, there will be presence of another octamer before octamer. What is the role of this H1 histone protein? Let's see. This is the H1 histone protein. So this H1 histone protein acts as linker. Here the DNA strand are there, and for the joining or for linking this whole structure, so whole structure के link करवार लगे, whole structure के बांधी करी रखवार लगे, there will be requirement of our H1 histone protein. So this is the H2A, this is H2B, and this is your H3, this is H4, and H1 histone protein. Okay, and around that octamer there will be coiling of the DNA. Now this whole structure which is formed here, having the octamer and H1 histone protein with the DNA, that is called as what? That is called as our another structure that is called as nucleosome. So this structure which is formed here, we gave a term. This whole structure now called as nucleosome. Okay. तो देखो nucleosome कहा के कहूछु. Nucleosome कहा के कहूछु. Nucleosome that is having octamer plus H1 histone protein plus DNA. The DNA that coil around the octamer and the H1 histone protein, that is also called as, the H1 histone protein is also called as linker protein. This is also called as linker protein. Okay. So this structure that is called as nucleosome, and this nucleosome again here there will be formation of another octamer. Suppose this is another octamer. Around that octamer again this DNA will coil, and again after 1.75 coiling again that will remove. काना बा देखो DNA फिर आसवा, DNA फिर आसिकरी octamer around रे coil हवा, DNA आसिकरी octamer around रे फिर coiling हवा 1.75 coil, 1.75 coiling for a फिर गुटे octamer आसवा, another octamer will be there. This is another octamer, and again the DNA will bind, and so that there will be formation of a chain of nucleosome. So here there will be requirement of our histone protein. There will be requirement of H1 histone protein in every octamer. So here H1 histone protein, this is H1 histone protein. Okay, so after several coiling around the number of octamers, there will be formation of number of nucleosomes. That means a chain of nucleosome will form, and that chain of nucleosome is now known as chromatin, or that is also called as nucleosome fiber. Okay, let's see the another sheet. Okay, I will share this. So how it will look? Suppose this is one octamer. In that octamer there will be presence of your H2, H2B, H3 and H4. This is another octamer. This is another octamer. This is another octamer, and this is another octamer, suppose. And let's see the DNA. The DNA will coil, again it will coil, again it will coil, again it will coil, and that. And this process will going on repeating. This process ta repetition haba, and after repetition, then you have the structure formation halla. This structure now called as nucleosome fiber. This structure is called as what? Nucleosome fiber. Okay. देखो DNA जत्ते वाला coil है जो DNA जत्ते वाला coil है गला coil हला पोरे हमर DNA रो length थिला DNA रो then our length thila length ta kana kari gala that go on decreasing, length ta kana kari jo decrease kari jo, but DNA and then structure formation ala DNA ru jetale nucleosome fiber formation ala nucleosome fiber formation is ala pore yahade jen our structure formation ala nucleosome fiber itar jodi ame width dekhochu, jodi itar width jodi dekhochu, width और जन उसार देखो उसार टा काना करे जिबा that will go on increase तार उसार टा increase करे जिबा तार width टा increase करे जिबा but तार जन length थिला बहुत लंबा जन thread थिला से लंबा thread टा काना करे जो छे लंबा thread टा धीरे धीरे compact हेतल जाउ छे धीरे धीरे size टा तार length टा धीरे धीरे कमी जाउ छे but तार जन width टा छे width टा धीरे धीरे काना हेतल जाउ छे increase करी जाउ छे. So इतर जन width जदि देखमा यहाते इतर width जदि देखमा width आसु छे 10 nanometer. इतर width आसु छे केते 10 nanometer. We will discuss that. So before we are going to that condensation process, this nucleosome fiber is nothing but our chromatin. ये जो nucleosome fiber अच्छे इता क्या मैं कहूँ मैं काम लिख रही chromatin fiber बोली करी कहूँ. ओके तो क्रोमाटिन फाइबर काना है क्रोमाटिन फाइबर होचे हमर डीएनए जतले कॉइल होचे H1 हिस्टोन प्रोटीन अराउंड रे डीएनए जतले कॉइल होचे H1 हिस्टोन प्रोटीन हिस्टोन प्रोटीन अराउंड रे दैट इज योर ऑक्टामर अराउंड रे सो दैट नंबर ऑफ न्यूक्लियोसोम फॉर्मेशन होचे से न्यूक्लियोसोम गोटे चेन के कहूचे हम काम लिखरी दैट इज कॉल्ड एज योर क्रोमाटिन फाइबर बोलिकरी कहूचे. ओके लेट्स सी स्टेप बाय स्टेप. सपोज this is the DNA. Okay, the width of the DNA that is 2 nanometer. Okay, मन रखवार पड़वा DNA रो width केते रहउचे the width of the DNA that is 2 nanometer. This is DNA, and this DNA that coil around the nucleosome it will make this structure. DNA जते वाला coiling होउचे that will make this structure. जति तक हम रिप्रेजेंट करू छु केनदा दुसे जस्ट गुटे सुतारे मोती माने गुथि हला जंदा दिस it will looking like this. माने a beaded like structure will appear on a string gote रस्सी रे bead जेंता मोती माने थिसी सेंता appearance होचे so ई structure के कहूचो nucleosome fiber the chromatin fiber बोली करी कहूचो so here next level coiling so this is the fast level coiling this is the fast level coiling. After fast level coiling there will be formation of nucleosome fiber. These are the octamers. So here the DNA that will bind, okay, and now this structure which is formed here that is known as our nucleosome fiber, and the nucleosome fiber have the width, the width increase kari jauche, the width that become 10 nanometers, and that is your nucleosome fiber, or we write this as chromatin fiber. Okay, then after that it undergoes coiling, another level of coiling. It undergoes another level of coiling. Next, that is your second level coiling. In the second level coiling, what will happen? Here this nucleosome, I am just drawing this structure in a very small structure. I have shown this बहुत गोटे छोट स्ट्रक्चर देखुछ बट दैट इज वेरी लॉन्ग. यह जेन स्ट्रक्चर देखुछ न्यूक्लियोसोम इटा में बहुत छोटे पार्ट काटे छे बट दिस इज बहुत लॉन्ग अच्छे, बहुत लंबा अच्छे. से जेन लंबा स्ट्रक्चर अच्छे से लंबा स्ट्रक्चर डीएनए भी जेन काटे छे डीएनए भी वेरी लॉन्ग स्ट्रक्चर बट ई जेन लंबा स्ट्रक्चर अच्छे ई लंबा स्ट्रक्चर टा मेजर एक्सिस अराउंड रे कॉइलिंग होचे. नाउ इट विल कॉइल अराउंड इट्स ओन एक्सिस. इन द सेकंड लेवल रे कॉइलिंग रे काना होचे इट कॉइल अराउंड its own axis, tar निजर axis around the coil ही करी, that will make another structure, another compact structure, and this compact structure that is known as what this, and now this width will increase, and that is having the 30 nanometer width, and this 30 nanometer width structure, now this is called as solenoid. Solenoid, and here we can find the nucleosome in that part. These are the nucleosomes. So DNA or that nucleosome fiber undergoes next level coiling, and that coil around its own axis. जब निजर axis around coiling होचे तो इटा के कोई से intra coiling. This is called as intra coiling. Okay. But इना देखो coil कहाँ around रही थिला ई जगह रे हम जेतेले देखुतला in case of war ई जेतेले देखुतला histone protein around coiling होतला so that अरे के inter coiling बोले इर कहमा. But इना निजर axis रे निजे coil होचे just like the spring जंदा गुटे strand थिसे निजर axis रे निजे coiling ही थिसी. Now it will appear just like a spring. तो इस structure के कहूँ हम काम बोले करी solenoid बोले करी कहूँ. And this solenoid undergoes next level coiling. Now this will undergoes next level coiling. Or that is the third level coiling. After third level coiling, now it will appear, it will coil, several coiling will be there. It will coil around its own axis, and that coiling is repeating here. आहुरी धीरे धीरे coiling होसे, बहुत coiling जेते बेले होसे, there will be formation of a compact structure, and इन जे देखमा there will be several processes रे हमर काना थी, there will be presence of our nucleosome. Here there will be presence of the nucleosome. Okay, DNA ta coil is nucleosome around रे, so these are the nucleosome. आहुरी स्ट्रक्चर टा काण हेते जाउचे कॉम्पैक्ट हेते जाउचे छुआ हेते जाउचे तार विड्थ ता काण हेते जाउचे विड्थ ता आहुरी इंक्रीज हेते जाउचे एंड दिस स्ट्रक्चर that is having the 300 nanometer and this is called as what? Super solenoid. This is called as super solenoid. And this super solenoid undergoes next level coiling or third level coiling, that will undergoes third level coiling, and after third level coiling आहुरी जितेला compactation होचे there will be formation of this structure. We call this structure as the chromosome or chromatid. Here we can find the chromatid. Wait. When we see this, more coiling will be there. There will be more coiling. So this is fourth level coiling. Sorry, this is the fourth level coiling. This is fourth level coiling, and after fourth level coiling there will be formation of the compact structure of the DNA. That structure is now having the width of 700 nanometer and this structure is called as what? Chromatid. इटे गुटे अच्छे बोली दैट इज कॉल्ड एज क्रोमाटिड बट डीएनए जेतेले रेप्लिकेशन हे जबा डीएनए रो रेप्लिकेशन हे आगनो रेप्लिकेशन हे जे धन्य एटा होचे सिंगल स्ट्रक्चर क्रोमोसोम क्रोमाटिड सॉरी क्रोमाटिन नहीं दिस इज द क्रोमाटिड दिस इज द क्रोमाटिड ओके एबे 2 मिनिट पर कटि जबा तो आगे रिजनिंग कर दबा टॉपिक टा के सारे दबा ओके नेक्स्ट जेतले डीएनए रो रेप्लिकेशन होचे डीएनए रो रेप्लिकेशन हिसाला परे डीएनए रेप्लिकेशन हिसाला परे we can find this type of structure and इस structure हमर काना पेजन अच्छे there is DNA okay तो DNA केतेटा अच्छे ना DNA दुईटा set of DNA अच्छे but you know हमर DNA गुटे थिला but you know this is one DNA this is having another DNA two copy of DNA will form and this structure that is called as chromosome this structure is called as what chromosome and the chromosome have the width of 1400 nanometer. So this is the process or this is the process of DNA packaging, how this DNA packaging will occur and after that DNA packaging this 2 nanometer structure is converted into 1400 nanometer structure. Okay, size of the DNA will decrease here but the width of the DNA will increase here and that structure which is formed here that is called as chromosome. Okay, तो क्रोमोसोम रो स्ट्रक्चर काना रहउचे क्रोमोसोम जे देखुछु क्रोमोसोम काहा के नेगरी बनला क्रोमोसोम दैट इज ओनली मेड अप ऑफ यदि क्रोमोसोम देखला क्रोमोसोम रे हमर काना अछि यदि क्रोमोसोम रे देखुछु क्रोमोसोम रे ओनली देयर इज प्रेजेंस ऑफ द हिस्टोन प्रोटीन एंड द डीएनए यदि द बाहर कर दमा देन वी कैन फाइंड द डीएनए हियर ओके दैट मींस द क्रोमोसोम दैट इज मेड अप ऑफ व्हाट द डीएनए and DNA रो किछि पार्ट के हमें कहसु हमें काम वाली कि जीन वाली कि कहसु we will discuss and the genetics part this is not concerning in your cell biology part so this is the process by which the DNA undergoes several level of coiling and recoiling and after several level of coiling and recoiling it will make this structure